So now we're going to talk about humoral immunity. So humoral immunity. This is just going to be when we have things like B cells and antibodies. That these are going to be circulating throughout the blood to try to defend the body from any type of foreign substance or pathogen from entering into the cell. So this is going to be all strictly outside of the cell. It's not going to be we're not going to be killing off any cells directly like we did with the natural killer cells or the T cytotoxic cells. So let's go ahead and do some quick terms here. So we've talked about this in lab before, but we have what's called antigens. So antigens are just going to be, sometimes you'll see them abbreviated as antigens like this, AG. These are going to be foreign substances that elicit an immune response. Okay, so these are going to be different things. So it could be pathogens, it could be toxins, like poison. So these are going to elicit an immune response, and by doing that, these are going to cause the body to produce antibodies. So let's put here, it's going to lead to production of antibodies. So what exactly are antibodies? So antibodies, you'll see abbreviated as AB. These are also called immunoglobulins or IG. So these are going to be molecules that are going to be produced by plasma cells. So these are produced by plasma cells. And plasma cells actually, they come from B cells. They're derived from B cells. Fix my tape here. Plasma cells, but from B cells. Okay, so what's going to happen is the antibodies, remember in lab, when we drew them, they drew a representation like this. So antibodies, they're going to interact with antigen by binding to a specific epitope. So what is that? What does that mean? So an epitope, we'll just say we'll say it's S. aureus, right, just like from lab. And here's protein A. It's hanging off the surface of its cell. So put protein A. And there might be, lots of these little protein A's on the surface. So while we have antibodies that are going to bind to protein A, if they're anti-protein A, the part that it binds to, whether it's this part here, this part there, this part here, this part there, wherever, that's going to be called the epitope. So the epitope is going to be like the more specific pinpoint location where that antibody binds. So let's put epitope is part of the antigen. So the antigen might be considered as protein A if it's the whole protein, but the part that the antibody actually binds to is going to be called the epitope. So the epitope is part of the antigen to which the antibody binds. So maybe the antibody binds, this little spot right there. So it means on this one too, if this is protein A, it'd only bind that specific spot. All right, so we'll just say there, that's the example. All right, so what happens, what's the whole point of having antibodies even binding to different antigen? What does it matter? Why do we care? So different things can happen when antibody binds to antigen. So I'll just put here results of antibody binding to antigen. So we've already learned about one of these in class. We've learned that agglutination happens. So remember, when we have agglutination, all those antibodies are going to cross-link and clump up those bacteria to reduce the number of infectious units to deal with. So instead of, a macrophage having to go chase down 100 different, SREs, instead, all those antibodies clump them into just a few different groups, and it's like, sweet, here's a group of 30 SREs, and it can just, engulf them all at once. So anyways, when we have agglutination, this is going to reduce the number of infectious units to deal with. So another thing that can happen is when we have antibodies binding to antigen, we can get opsonization. So remember, we learned about that back in, I don't know where that paper is, back when we talked about the complement system. So remember when we had the complement system, we'll grab it here. Remember when we activated the complement pathway, 3 results happened. We had opsonization, inflammation, and then we had cytolysis, where we had that membrane attack complex form. Anyway, opsonization, if you remember, this is whenever the microbe or the pathogen gets coated, and then the macrophage or whatever is able to more easily phagocytose that pathogen. It's kind of like little handlebars to grab onto, or like little sticky tape or something, that it makes it easier for the phagocyte to engulf the pathogen. So opsonization occurs. So instead of, though, with complement protein, we're going to coat the pathogen with antibody for easier phagocytosis. And then we can also have the activation or activate the complement system. So just like before, remember, in the classical pathway, remember there's 3 ways to activate the complement system, but in the classical pathway, antibodies can bind to an antigen, thus starting the complement, or beginning the complement system. So when we activate the complement system, through that classic pathway, we're going to get inflammation, cell lysis, and even, further opsonization whenever those microbes get coated with the complement proteins, as well as, the antigens or the antibodies. So another thing that can happen is we can get antibody dependent cell-mediated immunity. So in this is just a fancy way of saying that we're going to have antibodies that attach to a target cell. So usually this is going to be a large parasite. So a lot of times like worms. And so whenever like this worm gets coated with antibody, this is going to signal for destruction by different immune cells. So things like the macrophages, eosinophils, and remember we just talked about the natural killer cells. These all kind of tag team and, shoot this parasite with lytic proteins to try to destroy it that way. So lastly, we also have what's called neutralization. So this is the last effect. So a neutralization, let's say we got a poison that entered into our bodies. So, or here we'll just say a virus. Better draw a virus. So anyways, let's say we have a virus that enters into our bodies, and what does the virus have to do in order to infect one of our cells? It has to attach to it, right? So what if our antibodies coated this virus and the virus was no longer able to attach to the cell? So if it was no longer able to attach, then that means that the virus would be able to, not infect the cell. Our cell would stay safe, and then that's what's called neutralization. So that's whenever antibodies block adhesion or, attachment to of the bacteria. It can also be a virus, even a toxin, like a poison or something. It blocks that to binding to our cells. So, right.